My dear fellows and my friends and my colleagues and residents, all medical officers, all who are attending these type of tutorials, although these type of tutorials are called very dry tutorials because it belongs to infections and the disease, and we are discussing actually the clinical microbiology, the application of the microbiology in medicine. That it means that you have to know, if you are appearing in the fellowship examination or in the higher examination, then you have to know everything about it. Otherwise, there will be no forgiveness. You have to face again that examination. I'll say one should know these things, although it is in third year MBBS, they told about us about the microbiology, that is bacteriology, and the parasitology, and it is unfortunate for us that we are not able to remember the past. Actually, the good past can be remembered. The bad past usually will be deleted in your mind. In most of the persons, these pharmacology subjects and microbiology is concerned, or jurisprudence, forensic medicine, and the preventive medicines, they are very dry subjects, and people do not like it, and most of them, the students, but they are very important subjects. Remember, it is and their application in the medicine, or in the, you can say, in the clinical side, that make you strong to have the proper diagnosis and management. So I have previously discussed about 7 or 8 tutorials, we can say, that is the gram positive cocci. I have covered most of these cocci as far as is concerned. Today I want to discuss the something, first of all, about the introduction of the gram positive and the gram negative bacilli, because only the remaining things which I have to give is the pneumococci and gonococci, which I will take later on. But before going to the bacilli or gram positive, rather bacilli or gram negative bacilli, this is the homework for you, because you have to remember these positive and the gram negative bacilli. Remember, they have been asked. For example, my colleague who was appearing in the MRCP examination, it is in probably in 1998, he was asked in the table viva that about the leptospirite, and they asked about something about the other food poisonings related bacilli, which are the gram positive bacilli involved in the food poisoning, and he was not able to answer. And they also asked one question at that time, which is the most important gram positive bacillus which is important in the food poisoning in pregnancy. Now, there were 2 questions which were asked in 1998 or 99, and the candidate was not able to answer because he thought that this is not important. Everything is important in the medicine. I am Professor Syed Ali Azhar. I am representing actually Jinnah Sindh Medical University. Now, basics of the bacteriology is that the gram positive bacteria that give a positive result with the gram stain. There is the basic stain of the gram, which is traditionally used for the classification of the bacteria into the 2 broad categories according to the type of the cell wall, because the cell wall consists of, the bacterial wall. There is a lipid layer which is a very important part of the absorbent of that gram staining. And the gram positive bacteria take up the crystal wall stain used in the test, because this is a mixture of the crystal violet and other agents which they used in the test and then appear to be the purple color. They are purple color when seen through the optical microscope, low power microscope, and they can be catched easily. This is because the thick peptidoglycan layer in the bacterial wall retains the stain after it is washed away from the rest of the sample in the decolonization stage of the test. Because first of all, you to stain it, dip it, and then you wash it after fixation. And if you see under the microscope, when you see this the basali or cocci, purple color, they are gram positive. Conversely, the gram negative bacteria cannot retain the violet stain, although they catch it, but they cannot retain it. After the decolorization step, the alcohol used in the stage degrades the outer membrane of the gram negative cells, making the cell wall more porous and incapable of retaining the crystal violet stain. So they will be washed with the alcohol also to confirm it. And their peptidoglycan layer is much thinner, and it is sandwiched between the inner cell and the membrane and the bacterial outer membrane, causing them to take up the counter stain. Other stain is the safranin or the fish sign. Stain is there in the gram. It will then counter stain with this safranin or the fish, and that is why they will appear red or pinkish in color rather than crystal violet. Although the color will be there, why? Because of the safranin and the fishin, which will take up by the inner cell membrane and the outer cell membrane, peptidoglycan layer will take it now. Despite their thicker peptidoglycan layer, gram-positive bacteria are more receptive to the certain cell wall targeting antibiotics, although they are thicker peptidoglycan layer. So they are more receptive and they are more sensitive to the action of the antibiotics acting on the cell wall, for example, penicillins. And then the gram-negative bacteria, due to the absence of the outer membrane, they lack the outer membrane thickness, and that is why it is difficult to act by those antibiotics which are acting on the cell wall. So they are not able to act properly. That's why the beta-lactam antibiotics, most of them are not good for the gram-negative bacilli. This is the concept why the gram-positive bacilli are sensitive to the drugs, like for example the penicillins or sulfonamides or fusidic acids or vancomycins, and in some of the macrolides even. They are sensitive because of they are acting on the peptidoglycan layer, thick layer they have. Their receptors are there on the membrane, and they definitely act on the cell wall and destroy their cell wall, and they are destroyed completely in the sense they are bactericidal in nature. While in the gram-negative type of the bacteria, they cannot act because it is lacking in the membrane is not present, outer membrane, and there are no receptors are there. So they cannot bind on the receptors, and they cannot bind on the receptors. How can they act? They cannot act. So they are poor on action about the penicillins are concerned against the gram-negative organisms are concerned. For example, gonococci is gram-negative. It is the meningococcus which is gram-negative. We will discuss. And gram-negative bacilli, many of them are present. Escherichia coli, Pseudomonas, proteus, salmonellas, they are not good for these penicillin groups of drugs. This is the concept actually, and they are asking, when your examiner is very knowledgeable, they can ask anything which is related to the matter. You have to remember it, definitely. Why they are looking crystal violet? Why gram negative are on the optical microscope are looking red or pink? Why? You have to answer by this slide. This is very, very clear here. It is definitely crystal white, crystal water clear concept of the. And why the certain antibiotics are not effective against the gram negative organisms? Answered in this slide. This is a violet stain gram positive cocci and pink stain gram negative bacilli. Both are seen here. Seen here. The chains, it is gram positive. Chains are seen here. And gram negative bacilli here, they are pink. It is crystal violet. And this is the bacilli group, which is stained because of the fusion, staining red. And the crystal violet, because of the crystal violet, it is appearing definitely, and the outer membrane pick it up very easily, and they are appearing as the violet colored, pink, slightly violet color, you can say, purplish violet color. Here absolutely purple color are there in the gram negative organisms. Now the red shaped gram positive Bacillus anthracus bacteria in the cervical fluid is demonstrated to understand. Now from the round, it is the round white blood cells. These are the blood cells. Okay, in between the, see the rods. These are the rods, and these rods are crystal violet in color. Because it is crystal violet in color, it is gram positive Bacillus inside the cerebrospinal fluid, and here they are demonstrating the Bacillus anthracus. The characteristic features of these gram positive bacteria in general, they have got the cytoplasmic lipid membrane, and they have thick peptidoglycan layer, and teichoic acids and lipids are present, forming the teichoic acids, which serves as the chelating agents, and also for certain types of adherents. Adherents are there. Chain will be formed like in this fashion. The peptidoglycan chains are cross-linked to form the rigid cell wall by bacterial enzyme DD transpeptidase. A much smaller volume of the periplasm, is the fluid which is present, than in the gram-negative bacteria. Only periplasm means that the fluid between the two membranes of the plasmalemma or cell wall is very small in volume than related with the gram-negative bacteria, when they will stain not with this crystal violet, but they stain with the fusion. Now, only some species have the capsule, usually consisting of the polysaccharides, capsulated organisms. Also, only some species are flagellates, and when they do have flagella, have only two basal body rings to support them. Whereas the gram-negative have four basal bodies. Both gram-positive and gram-negative bacteria commonly have a surface layer called an S layer, surface layer. In gram-positive bacteria, the surface layer is attached to the peptidoglycan layer very tightly. Gram-negative bacteria, this layer that is called the surface layer is attached directly to the outer membrane. Specific to the gram-positive bacteria, again, is the presence of the teichoic acids. Now, teichoic acids in the cell wall. Some of these are the lipoteichoic acids, which have a lipid content of the cell membrane that can assist in the anchoring the peptidoglycan. That is why they retain the stain. and their action of those antibiotics are, they are very positive to kill this bacteria. They- and they become bactericidal in this sense. Now, gram-positive and the gram-negative cell wall structure are explaining here. Gram-positive, you see the plasma membrane here, and then we have got the peptidoglycan. This is one, and this is the periplasmic space, whereas space has got very small amount of fluid. On the other hand, the gram-negative cell wall, you have got the C structures. You see, there is a plasma membrane, and the periplasmic space contains the fluid, the higher amount, definitely higher amount, and it got then thick peptidoglycan layers here, and then they have got outer membrane, liposaccharide and protein layer here outside. Because the gram stain is sensitive to this peptidoglycan, it is outside, the stain directly. Here it is very difficult to reach it. You have to, postmortem it, the outer layer, outer membrane, for the crystal wallet to stain the peptidoglycan. But they will stain on the other hand, with the other stain, like fusion stain. And it becomes purple color, therefore. It is crystal violet in color, it is purple color. The structure of the gram-positive cell wall is explaining in detail here. They have got how much proteins are present, how much liposaccharide is present, etc. In the gram-positive bacteria for their characteristic staining and their characteristic mode of action, and therefore characteristic, the antibiotics to attack on the walls. Now, classification overall of these gram-positive or gram-negative organisms are the gram-positive. Either it is cocci or it is bacillus. Cocci occur in chains. They are a rounded shape and oval shape bacteria. The cocci may be gram-positive, it may be gram-negative. Gram-positive I already discussed. Gram-positive or gram-negative. Another important point which is here, you see, is whether they are catalase positive or catalase negative. If they are catalase positive, now first of all, example, please give me the example of the gram-positive cocci. They are Staphylococcus and Streptococcus pneumoniae. Example of the gram-negative cocci is concerned, they are the gonococci and the meningococci. These are the examples. On the basis of the catalase positive or negative, the Streptococcus will be classified further. The Staphylococcus and Streptococcus, whether they are Staphylococcus or Streptococcus, further division based on the catalase enzyme is present or not. If they are catalase positive, then do the coagulase test. If it is Staphylococcus coagulase negative, then their pathogenesis is different. But it is negative, for example, the Staphylococcus saprophyticus, Staphylococcus epidermidis, etc. So they are coagulase negative. But Staphylococcus pyogenes, for example, they are coagulase positive. It is a very penetrating and invasive organism. In the same fashion, the Streptococcus will be classified. It is catalase negative. These are catalase negative, not positive. But about the coagulase is concerned, it is not depending on the coagulase reaction. It depends upon the hemolysis, whether alpha hemolytic or beta hemolytic or it is gamma hemolytic. Or they are dividing into what is called Lancefield grouping. A or B or C or D or E, etc. Now here is the beta hemolytic. Hemolysis means hemolysis have been done, clear. It's clear, all is gone. Streptococcus, for example, Pyogenes, group A bacterium, sensitive, and it has got a lot of mode of action and has got a lot of features. And Streptococcus agalacti, it is group B, Steinfeld grouping B, and it has got a very important action as far as concerning, I'll say the endocarditis is concerned. And about the gamma hemolytic organisms, they are enterococcus organisms. They also cause these type of lesions. They can cause pneumonia, they can cause endocarditis, fecalis, enterococcus These are organisms which are gram-negative organisms actually, and they are gram-positive organism, although gram-positive organism, but they are catalase negative organism. Hemolytic is gamma, means complete, they don't touch it. The gamma means they do not hemolyze even. So, the pathogenicity depends upon this enterococcus or the enterococcus faecalis or enterococcus faecium. These are the important agents in some of the cases of the vegetative endocarditis. And another one is the alpha hemolytic type of the reaction given by the streptococcus. For example, the streptococcus pneumoniae here coming here, and the octokin sensitive bile soluble capsule, etc. They are equally positive. I don't want to discuss further because it is definitely a very general bacteriology which I am discussing here. Just to remind you that there are alpha hemolytic streptococci, Streptococcus pneumoniae, Streptococcus viridans, it is mutants or sanguis, or these are important organisms, of the what we call as the vegetative endocarditis is concerned. Many of these organisms are also causing the pneumonia. Many of the organisms, they are causing the abscesses. They are the second invaders also, and the skin infections and the urinary tract infections and the abscesses inside. Abscesses like, for example, the retropharyngeal abscess, etc. These are the very important group of organisms. As far as the bacilli are concerned, gram-positive bacilli are concerned, they are Corynebacterium and Clostridium and Listeria and Bacillus. Bacillus cereus or Bacillus anthracus. Listeria, Clostridium, Corynebacterium, 1, 2, 3, 4. 4 organisms will be discussed. I am sure about it that most of the candidates who are appearing in the FCPS Part 2 clinical side, practical side, if they are asked about this, they forget actually, because they are not expecting that the examiner can ask this. So please, if you want to give this examination, you have to prepare A to Z. Now, importance of the outer cell membrane in the bacterial classification, I have already said yes. Although bacteria are traditionally divided into two main groups, the gram-positive and negative, based on their ground state retention property. Now, this classification is very, very old, but it is ambiguous. It refers to 3 distinct aspects: staining result, envelope organization, and taxonomic group, which do not necessarily coalesce to some bacterial species. Gram-positive and gram-negative staining response is also not a reliable characteristic, as these two kinds of bacteria do not form the phylogenetic coherent groups. आप ये नहीं कह सकते कि अगर ग्राम पॉजिटिव है तो वो एक खास किस्म का पैथोजेनिसिटी कॉज करेंगे, पैथोफिजियोलॉजी कॉज करेंगे, स्पेसिफिक लीजन्स डेवलप करेंगे, आप ये नहीं कह सकते. So its basis lies in the marked difference in the ultrastructure and the chemical composition of the bacterial cell wall. As soon as the electron microscopy has been developed, they actually studied why they are more invasive, why they are novel type of organisms, marked by the absence or presence of the outer lipid membrane. This is a membrane they have studied in the under the electron microscope, and from there they actually have taken their knowledge which bacteria are more invasive bacteria and which bacteria are not very invasive bacteria, etc. All gram-positive bacteria are bounded by single unit lipid membrane, and generally they contain a thick layer 20 to 80 nanometer of the peptidoglycan responsible for retaining this gram stain. A number of the other bacteria, they are bounded by single membrane, although they are single membrane, but stain gram negative due to the thick lack, due to either lack of the peptidoglycan layer in them. The peptidoglycan layer is not very thick. For example, in Mycobacterium pneumoniae, although Mycobacterium pneumoniae is a peptidoglycan layer which is there, but it will not stain with the very weak stain. It has an ability to retain the ground stain when you wash out with water and alcohol because of the cell wall composition. Also show close relationship to the gram-positive bacteria. Although their mode of action and they do it, the mycoplasmas are concerned. And remember the questions in about 20 years, 3 or 4 times they have asked in the theory examination about the mycoplasma, and I remember in my 12 or 13 years of clinical examiner in FCPS Part 2, and there are the examiner have asked about in the differential diagnosis, and the candidate put the mycoplasma also as atypical pneumonia. So they started about the mycoplasma, what are mycoplasmas and etc. about them, how they act, where the portal of entry and so on, and why they are so invasive and what is the pathogenicity. If you know these things, then you can understand they are gram negative, although their property of the is very mimicking the gram-positive bacteria. In contrast to the gram-positive bacteria, all typical gram-negative bacteria are bounded by a cytoplasmic membrane and an outer cell membrane. And they contain only a thin layer of the peptidoglycan between these membranes. The presence of inner and outer cell membranes defines a new compartment in these cells, the periplasmic spacer or the periplasmic compartment, where the fluid is there. These bacteria have designated as Didymium bacteria. The distinction between the monoderm and the Didymium bacteria is supported by the conserved single signature indels in a number of the important proteins like DRAC or GOL. Of these two structurally distinct group of bacteria, the monoderms are indicated to be the ancestral based upon a number of the observations, including that the negative bacteria are resistant to them. It has been proposed that the outer cell membrane in the gram-negative bacteria in the derms have enveloped as a protective mechanism against the antibiotic selection. They have got protective effect, DADMs, gram-negative bacteria. Pressure and some bacteria such as the Deinococcus, which is stained gram membrane, gram stained membrane as intermediate in the transition between the monoderm, gram-positive, and the diderm, gram-negative bacteria. The diderm bacteria can also further differentiated between the simple diderms lacking lipopolysaccharide and the archetypical diderm bacteria, where the outer cell membrane contains the lipopolysaccharide, and diderm bacteria where the outer cell membrane is made up of the myomarcloric acid. Now these are the substances, actually, inherited substances are acting as, if they are detached from the walls, they will act you as a pathogens and act on the hypothalamic and they will cause fever. It is also described where I was discussing the fever in my, I think, sixth previous lecture. The structure of the peptidoglycan composed of N-acetylglucosamine and N-acetylmumaric acid. These are the two things. Mumaric acid, these are the fumarate or mumaric acid. These are these substances which are very sensitive for the receptors in the temperature regulation, disturbing the hypothalamus, anti-rise and anti-fall centers, and it cause fever. The pathogenicity is as concerned. In the classical sense, 6 gram-positive bacteria are typically pathogenic humans. What? 2 are Staphylococcus or cocci, and also Staphylococcus pneumoniae. The remaining organisms are bacilli, rod-shaped, and can be subdivided based on the ability to form spores. The non-spore formers are Corynebacterium and Listeria, monocytogens. There is Bacillus anthracus, and Clostridium produces the spores. They produce the spores. They are spore formings. In this way, they are distinguished and they are culturing. The spore forming bacteria can again be divided based on the respiration. So, Bacillus anthracus is a facultative anaerobe. It will require very less amount of oxygen. While the Clostridium is obligate anaerobe. It is anaerobe, obligate. Very, very, very small amount of oxygen is needed. Facultative anaerobe, which is available oxygen, is sufficient for the growth. For the Clostridium is concerned, it will need definitely anoxic environment where they will grow. So it is based upon this they are subclassifying and subclassifying during the culture of which are presented to them, the clinical microbiologist. They culture it and they will specify you that it is the Corynebacterium or the Listeria or it is Bacillus anthracus or it is Clostridium, etc. This is how they do it. The mechanism is there. The spore forming or non-spore forming. And about the cocci are very clear. I have said that the Streptococci and the Streptococci and Pneumococci, which I have discussed previously, I think so. Now, bacterial transformation, it is a very interesting theory, and that it can be transformed. One structure is not fixed. This is called the horizontal gene transfer, in which the oxygen is genetic material passes from the donor bacterium to the reception bacterium. The other two processes being conjugation transfer of the genetic material with the two bacterial cells in the direct contact, and the transduction, the injection of the donor bacterial DNA by the bacteriophage virus into the reception host bacterium. In transformation, the genetic material passes through the intervening medium and uptake is completely dependent on the reception bacterium. That is why their characteristic changes. वो कहते हैं ना म्यूटेटेड हो जाता है, तो म्यूटेशन हो रहा होता है इसमें. आप उसको एक साल से मार देंगे लेकिन वो दूसरी तरीके से पैदा हो जाएगा, जैसे वायरस में होता है. इसी तरह यहाँ भी हो रहा है. As of 2014, about 80 species of the bacteria were known to be capable of transformation. About evenly divided gram-positive and gram-negative bacteria, the number might be overestimated since several of the reports are reported by the single papers. The transformation among the gram-positive bacteria has been studied in the medically important species such as Streptococcus pneumoniae and Streptococcus mutans, or Streptococcus aureus and Streptococcus sanguinus, and in gram-positive soil bacterium like Bacillus subtilis and Bacillus cereus. And in them, The thing is this, that they are transferring their characteristic features now. So the mutations occur in these bacteria also. One time a bacteria was a gram positive, now it is gram negative. At one time it is intermediate. At one time it is sensitive to the penicillin, now it is not sensitive to the penicillin. So bacterial transformation is one part in which the antibiotic resistance against the bacteria can be explained. So this is actually the basic knowledge which I have given. It is not a very high flow lectures. It is very, I will say, basics which are given to you to understand the bacteriology, in the sense that if you are asked a question, then you are able to answer it properly. That is my aim of this presentation. And I am a clinician, I am not a clinical microbiologist. But how they are doing it, you should know. I am very thankful that you have tolerated this type of tutorial, and we will see inshallah more in the coming days. وما علينا إلا البلاغ. السلام عليكم ورحمة الله وبركاته.